Good afternoon, everyone. First of all, let me thank you all for giving me this opportunity today. My name is Koichiro Majima from Nagoya University, Japan. I am honored to have this opportunity to give this presentation in such a prestigious conference. Let me start by saying a few words about my own background. I was born in Japan. My mother loved traveling and took me to Europe again and again, even when I was a newborn baby. Like these countries, Germany, Switzerland, UK, Italy. However, this is the first time for me to set foot on French soil. Why have my parents never brought me to France? My parents love The Little Prince by Saint-Exupéry, who walked in Lyon. A famous quote from this book is, "All grown-ups were once children, but few of them remember it." My parents said to me, "You don't truly understand the beauty of France until you've grown up, and you won't remember it when you become a grown-up." Therefore, they never brought me to France. And now here I stand, embarking on the process to be a grown-up. All animals begin as single zygotes, maturing into fetuses, neonates, and eventually grown-ups. I'm interested in this process, differentiation. All cells have the same genetic DNA. However, it is true that all grown-ups were once zygotes, but none of them remember it. Now, let's move on to research. Single-cell RNA-seq is a powerful tool for differentiation studies. In single-cell RNA-seq, tissue is dissociated, single cells are lysed, and their RNA is sequenced. Single-cell RNA-seq offers various analyses, but time information is lost due to cell destruction. Time information is lost due to cell destruction. There are the major methods for reconstructing temporal information used in differentiation studies: RNA velocity and Larry. RNA velocity uses splicing kinetics, predict the direction of RNA expression state transitions in a short period. However, it has limitations in accuracy predicting long-term cell evolution. Larry introduces genetic barcoding and provides time series tracking. However, it captures only sister relationships in sparse observation time points and cannot trace ancestral and intermediate cell states. No method has yet been developed to bridge between these two methods, short-term and long-term. Therefore, we developed Lineage VI. Lineage VI recovers cell states and expressions at single cell level at each time point. Our research focuses on these three key motivations. First, inferring dynamics and unobserved cell state at the single cell level. Second, recovering historical expression at each time point. Third, estimating transcription factor TF activity along cell trajectories, cell differentiation trajectories. Here is the overview of our method. In our research, we utilize single-cell RNA-seq and lineage tracing data. We use a neural network repeatedly to estimate the dynamics up to the ancestral state. As downstream analysis, by leveraging this method, we can perform continuous dynamics, historical gene expression, and TF activity along the trajectory analysis. Trajectory analysis at the single cell level and for each time point. We performed this study using Larry's publicly available dataset. In this dataset, barcode embedded at day 0. After that, on day 2, 4, and 6, some cells were harvested and performed single-cell RNA-seq. Our method, lineage PI, is VAE-based model. The VAE uses 2 neural networks to uncover data structure. The encoder maps data to a low-dimensional latent space Z, while the decoder reconstructs the original data. The latent space capture data features. We extend Z to reflect lineage information. Here is the generative model that our models assumes. We assume one common ancestor for each lineage, and an ancestor cell diverging to the number of cells observed in the initial transition, first step. Each cell changes its state in the latent space until the observed time. We assume that the observed data for each cell is generated from each latent state. This is the whole picture of our generative model. Next, let me introduce the variational inference of our model. Each observation data is mapped to each latent state at the observed time point. Our model conducts variational inference backward in time repeatedly and estimates transition dynamics in the latent space. For each lineage, it assumes that cell states are in the single state at time 0. This is the overview of how our model conducts variational inference. In training this model, we optimize the parameters of 3 neural networks using VAE-based LO, shown below. I will now proceed to explain the results. This slide shows the latent state UMAP display. The figure on the left shows the estimated cell state at each time point. The middle figure shows the time when the observation data was observed. The figure on the right shows the cell type annotation of the observed data. We use cell types that were originally annotated in previous studies. This is an animated view. We utilize training model and the day 6 observation data as input. It can be seen that cells gather in the undifferentiated region as time goes back. On this slide, we have a visual comparison of the inference and observation at each corresponding time point. It confirms that the inference is similar to the observed data. Then we compare our method with other methods. First, we compared our method with pseudo time and SC0, which are often used in the field of differentiation studies. These two methods on the right have failed to predict the location of progenitor cells and the direction of differentiation. Our model, shown in the figure on the left, performed better than pseudo-time and SC0 in predicting differentiation trajectories and progenitor cell state. This slide compares our method with Waddington OT, which uses the time point information. We evaluate the performance by interpolating day 4 and calculate the ratio using nearby observed time point of cells within the K neighborhood of the estimated day 4 cells. Our method, lineage VI, shown in the figure on the left, exhibits better performance than Waddington OT, depicted in the figure in the middle. One advantage of our method is that it allows us to recover expression at each time point. By checking marker genes, we observe that as time goes back, differentiation marker decrease, while undifferentiated markers increase. This suggests that accuracy of our model lineage VI. Next, we analyze progenitor bias by the following method. First, we extract barcodes where more than 80% of cells differentiate into one cell type. Second, we recover past expression using our method lineage VI. Finally, we detect differentially expressed genes, DEG, between cell lineage at day 0. This slide displays the results of the analysis on progenitor bias. DEGs include neutrophil and monocyte markers, shown in red square. CTST and GSTM1, not known as marker genes, may influence differentiation. It suggests possibly expression bias dependent on differentiation destination. Finally, let me explain TF activity along the trajectory, transcription factor activity along the trajectory. On the left figure, I presented the average differentiation trajectory of monocyte and neutrophil. Now, take a look at the figure on the right. At day 3 to 4, the distance between the trajectories dramatically increase, showing red arrow. Then we proceed with identifying TF's time differential activity. To evaluate TF activity, we assess the changes in downstream target gene expression at each time point using this regulation matrix. This slide shows the ID2, transcription factor ID2 activity, which exhibits high temporal variability. ID2 activity rises before neutrophils and monocytes move apart. I will now summarize this research. In the current situation, the analysis of progenitor and intermediate cell state is important. However, time information is lost due to cell destruction and sparse observation time points. With our model, lineage PI, we can overcome these limitations. It estimates continuous dynamics and unobserved cell states at single cell level. Moreover, it enables the restoration of historical expression and identifies TF activity along the trajectory of each cell. Additionally, lineage VI allows for the analysis of single cell differentiation trajectories. Thank you for listening.
Any questions? Yes.
So I see that different latent variables correspond to different time points. Is that something that you have to enforce during training, or do you just identify after training which latent variables are corresponding to which time points? Yeah, exactly.
You mean how deal with the time points, right?
How do you enforce that, like Z2 1 corresponds to day 2?
Oh, I see. Our model uses 3 neural networks, and yes, one is for embedded expression information in low dimensional space, and another is for inferring cell state transition dynamics. The other is for reconstructing. And let me show you. Sorry. So the model assumes that the progenitor cell in the first step divides into the observed number of cells, like this figure, and so the cells continue cell state change depends on the until the time point at which observed. So if the cell is observed at time point day 6, it continues the cell state change 6 times. And if the cell is observed in time 4, day 4, it conducts cell transition 4 times. So and assuming that it gathers in one place when time 0, that works well.